Welcome to Studious. Today, we are building the complete NeuroAtlas blueprint of one of the most fascinating and historically mysterious structures in the entire human brain, the pineal gland. Formerly known as the Epiphysis cerebri, or the pineal body, this small but extraordinarily significant gland has captivated scientists and philosophers for centuries. René Descartes famously called it the seat of the soul, the place where the mind and body meet. While modern neuroscience has moved well beyond that claim, what we now know about the pineal gland is arguably more remarkable than any philosophical speculation. It is a neuroendocrine transducer, a bridge between the nervous system and the endocrine hormone system that converts neural signals about light and darkness into hormonal commands that regulate your entire sleep-wake cycle and reproductive development. In this video, we will map its precise anatomical location, examine its structural architecture, trace its vascular supply, and understand its two critical physiological roles. Let's begin.
The pineal gland is a remarkably small structure, measuring only 5 to 8 millimeters in length, yet its influence on human physiology is disproportionately large. Its name comes from the Latin word pinus, meaning pine tree, because its shape precisely resembles a miniature pine cone. Clinically, it is also referred to as the Epiphysis cerebri, or the pineal body. In terms of its anatomical location, the pineal gland sits within the Diencephalon, the central division of the forebrain, also known as the prosencephalon. The Diencephalon is the deep core of the forebrain and houses some of the brain's most critical structures, including the thalamus, hypothalamus, epithalamus, and subthalamus. The pineal gland is specifically housed within the epithalamic region of the diencephalon, the posterior superior part of the diencephalon. On a mid-sagittal brain image, the pineal gland glows like a green beacon in the very center of the brain, sitting at the junction between the forebrain and the midbrain, in one of the most geometrically central and protected positions in the entire central nervous system.
To fully understand the pineal gland's anatomy, we need to map its precise spatial relationships with its immediate neighbors along 3 anatomical axes. On the anterior axis, the pineal gland projects posteriorly from the posterior wall of the third ventricle, the narrow midline fluid-filled cavity of the diencephalon. The pineal gland is essentially a posterior outgrowth from this ventricular wall, maintaining a direct anatomical connection to the ventricular CSF system. On the superior and dorsal axis, the pineal gland is located directly above the quadrigeminal plate, also called the tectal plate, which is the dorsal surface of the midbrain, containing the 4 colliculi, the superior colliculi for visual reflexes and the inferior colliculi for auditory reflexes. This proximity to the quadrigeminal plate is clinically critical because pineal tumors can compress the superior colliculi, causing Perronneau syndrome, a characteristic loss of upward gaze that is a classic clinical sign of pineal region pathology. On the lateral axis, the pineal gland rests precisely between the posterior aspects of the 2 thalami, nestled symmetrically in the midline groove between them.
The pineal gland is not a free-floating structure. It is anchored to the brain by a short stalk, and this stalk has a precise and clinically important internal architecture. The pineal stalk connects the gland body to the posterior wall of the third ventricle and contains 2 distinct flat connecting sheets called laminae, the Latin word for thin plates. The superior lamina connects the pineal body superiorly to the brain. It contains 2 important structures, the habenular commissure, a small but significant white matter crossing that connects the habenular nuclei of both sides, which are involved in processing aversive experiences, pain, and reward, and the habenular trigone, a small triangular surface area on the posterior diencephalon where the habenular nuclei are located. The inferior lamina connects the pineal body inferiorly. It contains the posterior commissure, a clinically important white matter crossing located at the junction between the diencephalon and midbrain. The posterior commissure carries fibers involved in the pupillary light reflex, and damage to it impairs the consensual light reflex. Together, these two laminae physically anchor the pineal body to both cerebral hemispheres and integrate it with important commissural pathways.
Between the superior and inferior laminae of the pineal stalk lies a specific anatomical space of considerable structural and functional significance, the pineal recess. The pineal recess is the specific empty space situated directly between the superior and inferior laminae, forming an extension of the third ventricle that projects posteriorly into the pineal stalk itself. This means that cerebrospinal fluid from the third ventricle directly contacts the interior of the pineal stalk, an unusual and functionally intriguing arrangement that exposes the pineal gland's internal environment to CSF signals. The pineal recess communicates anteriorly through two pathways. First, it directly opens into the third ventricle, the main ventricular cavity of the diencephalon, making it essentially a posterior diverticulum of the third ventricle. Second, it also communicates with the hypothalamic sulcus, the shallow groove running along the lateral wall of the third ventricle that marks the boundary between the thalamus above and the hypothalamus below. This CSF communication pathway is thought to be one route through which the pineal gland's melatonin output may influence CSF composition, and why pineal cysts visible on MRI must be carefully distinguished from true neoplasms.
Now we arrive at the most physiologically remarkable aspects of the pineal gland, its two endocrine functions, and the extraordinary anatomical exception that makes both of them possible. The first function is chronobiology and melatonin production. The pineal gland is the primary source of melatonin in the human body, the hormone that regulates the sleep-wake cycle, also known as the circadian rhythm. Melatonin synthesis and secretion are driven by the photoperiod, the daily cycle of light and darkness. Light signals detected by the retina travel via the retinohypothalamic tract to the suprachiasmatic nucleus of the hypothalamus, then through a multineuron sympathetic pathway to the superior cervical ganglion, and finally via postganglionic fibers to the pineal gland. In darkness, the pineal gland produces melatonin, signaling the body to sleep. In light, production is suppressed. The second function is reproductive regulation. The pineal gland is believed to inhibit the maturation of the gonads until puberty is reached, suppressing premature sexual development through its melatonin output. And here is the critical physiological exception. Unlike virtually every other brain structure, the pineal gland lies outside the blood-brain barrier, making it directly accessible to circulating hormones and blood-borne signals.
Having mapped the pineal gland's location and function, let us now trace its complete vascular supply, beginning with arterial inflow. The pineal gland receives its oxygenated arterial blood supply through a cascade of 3 vessels originating from the posterior cerebral circulation. The cascade begins with the basilar artery, the large midline artery formed by the union of the 2 vertebral arteries at the base of the brainstem. The basilar artery is the principal supply of the posterior brain, including the brainstem, cerebellum, thalamus, and occipital lobes. From the basilar artery, the blood supply ascends to the posterior cerebral arteries, the PCAs, which are the terminal branches of the basilar artery, wrapping around the midbrain to supply the posterior cerebral hemispheres, thalamus, and the deep diencephalon. From the posterior cerebral artery, fine branches called the posterior choroidal arteries arise, small but critically important vessels that supply the choroid plexus of the third and lateral ventricles, the thalamus, and crucially, the pineal gland itself. These posterior choroidal arterial branches represent the final link in the supply cascade, delivering oxygenated blood directly to the pineal tissue to support its continuous melatonin synthesis and secretory activity.
After oxygenated blood delivers nutrients to the pineal gland, the deoxygenated blood must drain efficiently out through the venous system. The venous drainage of the pineal gland is a convergent system. Multiple veins from different parts of the deep brain all converge at a single major collecting point. Deoxygenated blood leaves the pineal gland via the internal cerebral veins, a pair of deep cerebral veins that run along the roof of the third ventricle, collecting blood from the thalamus, caudate nucleus, choroid plexus, and pineal gland. These internal cerebral veins then converge with two additional deep veins, the basal veins of Rosenthal, which drain the deep temporal lobe, hippocampus, and medial midbrain, and the posterior mesencephalic veins, which drain the dorsal midbrain and the pineal region itself. All three of these deep venous tributaries converge to form the great cerebral vein of Galen, one of the most important deep cerebral veins, named after the ancient Greek physician Galen who first described it. The vein of Galen then drains into the straight sinus, a dural venous sinus running along the tentorium cerebelli, representing the final venous output from the deep brain. Pathological enlargement of the vein of Galen in neonates, vein of Galen malformation, is a serious neurovascular emergency.
We close with the complete pineal master reference, a 4-quadrant synthesis of everything covered in this atlas. In morphology and location, the pineal gland is also known as the Epiphysis cerebri, or pineal body, measures 5 to 8 millimeters in a cone shape, is located in the Diencephalon, specifically the epithalamus of the prosencephalon, and sits directly above the quadrigeminal plate. In microarchitecture, the stalk contains 2 laminae, the superior lamina carrying the habenular commissure and trigone, and the inferior lamina carrying the posterior commissure, separated by the pineal recess. In neuroendocrine function, the gland produces melatonin for sleep-wake cycle regulation, regulates puberty onset by inhibiting premature genital maturation, and crucially, operates as a notable physiological exception because it lacks a blood-brain barrier. In vasculature, arterial inflow arrives via posterior choroidal branches of the posterior cerebral artery from the basilar artery, and venous outflow from the internal cerebral veins merges with the basal veins of Rosenthal and posterior mesencephalic veins into the great cerebral vein of Galen, draining to the straight sinus. This is the pineal gland, tiny in size, extraordinary in function. Thank you for watching Studious.